Professor Medini from Toscana, he is Strategic Director of Data Science in Toscana Life Science Foundation, and he is interested in molecular biology, computational physics, and so on so far. And his lecture will be Artificial Intelligence for Research Development and Manufacturing of Biologics. Professor Medini, please, you can start now.
What I would like to share with you today is a few ideas. I will try not to go in any technical detail about how we have been using and how we plan to further use in the future artificial intelligence joined with biology and immunology to improve health using a specific focus on biologic products, which are monoclonal antibodies, vaccines, hormones. I'll spend a little bit on that. What I just wanted to share before I begin is the fact that artificial intelligence and every mathematical methodology that is developed to interact with experimental data, which are generated in biological laboratories or in medical experimentation, communicate among them through data, which is the most precious asset that is also the joining element between two communities that typically speak two different languages, and they actually communicate through this common language, which is data. And the outcome of all of this is better health for people. So let me first start by introducing a little bit, for those who may not be familiar with the term, what are biologic products? When we think of medical interventions or pharmaceutical interventions, pharmaceutical products are divided in two categories: chemical products, which are typically called drugs, and biologic products, which are called biologics. And the main difference between these two classes of products is that chemical drugs can be manufactured, synthesized by means of chemical synthesis. And this is very simple to do from a technical and an industrial point of view. And it's also very simple to standardize and very reliable as a manufacturing process. Once you have a chemical manufacturing line, you can use it to manufacture whatever chemical product you want. They are pretty similar one to the other. You see on the x-axis of this chart the molecular mass, and we are spanning a couple orders of magnitude for chemicals, from the 180 Daltons of aspirin, which is a small one, to the largest one that I know of, which is vancomycin. It's an antibiotic, which is just 10 times larger. Now, if we talk about biologic products, they are so complex from a molecular point of view that it's not possible to synthesize them chemically. To produce a biologic product, you need a living organism in your manufacturing process. And biologic products are so diverse, as opposite to chemicals. You start from more or less the same size. Enoxaparin is 1500 Daltons, more or less similar, but it's impossible to synthesize. It has to be produced by living organisms, up to hormones, monoclonal antibodies, vaccines like the spike SARS protein, or bacterial vesicles, or gene therapy through CAR T cellular therapies. It's a universe where the key element is complexity in understanding the functionings and being able to manufacture them. So how do we use data science in this world of very complex products? I'm taking here an example, which is a vaccine that has been initiated. The research and development work was initiated in 1997, and the product was finally approved in 2014 by the European Medical Agency and shortly after by the Food and Drug Administration, To give you an example of how we have been using mathematics and some artificial intelligence tools over the last 20 years. This is important to understand that there's nothing brand new here. We are not inventing something that has not been done in the recent past. So everything started at the beginning of the century with, in 1995, the first complete genome of a living organism. It was a small bacterium was determined. And the second one was actually through an agreement between the vaccinology research group in Siena that I work with, and the Oxford University and Craig Venter in the US, was the genome of this bacterium, which is called meningococcus, particularly the serogroup B of this meningococcus. The genome was sequenced with the idea that to determine what was the right component of the bacterium to be used as a vaccine, and the bacterium causes meningitis, which is a terrible disease, genomics would have given us the entire blueprint of the organism, which was true, and the technology was called reverse vaccinology. This is today the most established way of doing vaccines in general when you do vaccines for bacteria. And today we have developed that initial approach to use machine learning to do this antigen identification about 10 times better than we used to do in the early days 20 years ago. And the second area is when you start testing your components, you need to understand what's the impact of your product. And a very important element when you do infectious diseases is that your enemy is a species. It can be a viral species or a bacterial species or something like a plasmodium. The species is very diverse in itself. It's not a single individual that you are fighting against. We know today about variants of SARS-CoV-2. Not every SARS-CoV-2 is the same, even more so bacteria, which are hundreds times bigger and more complex than viruses. And so pangenomic was discovered, and today we use machine learning to understand the diversity of the population of the enemy that our vaccine has to protect us against. And then the clinical development phase came, and the necessity to develop assays that would predict what would have been the coverage of the vaccine towards this diversity. And initially a lab-based test was developed. It was called MATS, meningococcal antigen typing system. And today we use a genome-based replica of that assay, which is 1,000 times faster and uses mathematical analysis to establish what is covered and what is not. And finally, once we got into, after the clinical trials, into the worldwide population, we started using mathematical methods like synthetic controls or Monte Carlo machine learning and maximum likelihood inferences to improve the way we estimate the efficacy and the impact of the intervention at scale in the real population. So this is really just a snapshot of early discovery, clinical development, and implementation of vaccines using data science and artificial intelligence methods. One may ask, so every problem is solved, we know how to do things, it's all perfect. Not quite. This is not the case. The key word when we deal with biologic products is complexity. And I would like to spend the rest of my time with you today highlighting some of the challenges and some of the opportunities that we have in this space. First of all, the number one challenge, as I mentioned before, is manufacturing. And manufacturing of biologics is a challenge because due to the complexity, every new product that you design, you have to invent a new manufacturing process. So if you do a monoclonal antibody of some kind against COVID, and then you discover a better monoclonal antibody against COVID, you have to develop and adapt a new manufacturing process. Every time you do this, approximately $500 million goes in investment for them standing up the manufacturing line. And you understand this means that only big pharma can do this. So you can design a product only if you work in a company that can also manufacture and the product, and that can sustain investments in the $1 billion order to test your design. And this is really a limiting obstacle to the design of new biologic products. The result of this is very well known in the pharmaceutical industry. It's being called the E-Rooms law, by taking the Moore's law, reverting the order of the letters in the name, which basically means that every decade, the researching and developing a new drug costs 10 times more. So if in the 50s, with $1 billion of R&D investment, you could develop some 50 or 60 drugs, today, 1 billion is not even sufficient for one single drug. How do we go about solving this problem? I would like to touch 2 points. First, how do we remove the manufacturing bottleneck that I introduced? And then we take a step back, and once we removed that and we are free to discover more openly, how do we use artificial intelligence to understanding the language of immunity, which is at the heart of a number of indications, not only infectious diseases, obviously, but also autoimmunity, CNS, immuno-oncology, and many others. So we all witnessed the incredible success of RNA as a vaccine platform for COVID-19 vaccines, effectiveness more than 90%, best-in-class safety, super fast time to market, research and development in less than 11 months overall. And what everyone is convinced of is that COVID-19 was a demonstration, but it's not the single application that can be used for RNA as a platform. The key concept of RNA products is moving the manufacturing hurdle from these huge tanks that you see in the slide. These are fermenters that are enormous biological facilities where the manufacturing of biological products is performed. You move that inside the human body because you deliver the instruction to the human body. And so I manufacture my own vaccine inside my body, having been given the instruction with RNA. And these can expand to a number of very many indications that go beyond infectious diseases. How do we go about exploding that possibility so that RNA can become a platform that disentangles manufacturing from product design and makes product design free from the chain of every time developing a new manufacturing process? We are taking inspiration here from what happened in a completely different industry, in semiconductors, like far, far away galaxy. And what happened there is that since the first integrated circuit was invented in 1958, that industry has been exactly in the same situation that I'm describing here for biologics, because you could only design a new semiconductor-based product, a new circuit, whatever kind of product based on semiconductors. You could only design that if you knew also how to manufacture that. So you need to know quantum physics and how the parasite currents in the wafer would have dispersed if you had designed a certain semiconductor in a certain way. So it was exactly the same. And if you look at the graph here, you see the average number of new companies that were created every year as a proxy for the average number of new products that were invented. We are talking about worldwide an average of 3 companies per year created. Then something happened. In 1978-79, the famous Mead and Conway revolution happened. These 2 guys managed to separate manufacturing from design by abstracting the manufacturing process of semiconductor-based products. And after their work, you could go to a software and say, okay, I have a certain semiconductor design that I would like to manufacture. Is this manufacturable against some manufacturing process? And the software will tell you, yes, this process, but not this other process. And so the number of new companies and new products exploded by 2 orders of magnitude. And you could, at this point, have in the industry 2 models. One, the vertically integrated that was existing before, like IBM, or Hewlett Packard or Texas Instruments, where you do everything, design and manufacturing. But now you can also do design only and use the software to understand which manufacturing only company can manufacture your designs. This is what exploded the business. And this is what we are trying to do now by repeating exactly the same approach to the RNA manufacturing process, by developing manufacturing processes that are represented by an abstract layer of computerized and digital twinned processes that are controlled by an artificial intelligence engine that allows exactly the same kind of simulation and then manufacturing independently from the specificity of the process. Now, if we succeed in transforming something like RNA into a general platform, we have solved the problem of manufacturing, but we are left with the complexity in the life systems that we are dealing with at the molecular level. And I would like to give you a sense of how the science of complexity can help in understanding the fundamental mechanisms that we're dealing with here. And I will start from the example of the pangenome. The pangenome, that has been called the language of life, is a concept that we developed in 2005, when we basically realized that the rules that govern the diversity of genomes are exactly the same rules that govern the diversity of human languages that has been studied for many, many, many centuries by linguistics first and then computational linguistics. And what you see on the left of the graph here is a chart in log-log scale. It's a very famous chart from the end of the 70s by a computational linguistic hero that was Professor Heaps. You see on the x-axis the number of new words that you find as long as you keep reading new text corpora or books written in a certain language. No, I'm sorry. On the y-axis, you find the overall number of words. What you have on the x-axis is the number of books that you read. And you see that words and books are connected by a power law. So on a log-log scale, they are a straight line, which basically means that in a living language like English, for example, or Russian or Italian, the more you keep reading, the more new words you find. This relationship does not saturate. There's not a point, if you ask the question, how many books should I read to be sure that I know that I've encountered at least once all the words in a given language? There's no such number, because this is an open system. And this is because the language is alive. Every year, new words are created, new variations are created. And so this keeps expanding. This is exactly the same thing that happens with genomes. If you take a certain bacterial species and you sequence 1 individual, 2 individuals, 10 individuals, hundreds of individuals, what you find is that the number of genes pertaining to that species is not finite for some species. For those who have, like Russian or English or Italian, like living languages that have mechanisms of novelty and diversity creation. And this, if you want to develop a vaccine, for example, or a monoclonal antibody against that pathogen, it's a fundamental feature of that pathogen, because you understand that the diversity that you're dealing with is unbounded. And this has now become a discipline in itself. It's been applied way beyond bacteria to every living organism and in many different ways. What's the novelty here? The novelty here is what happened a few months ago with particularly the AlphaFold 2 breakthrough and the Rosetta Fold breakthrough that followed shortly thereafter. What we can do now with machine learning is predicting not the sequences like we do in the pangenome work, but the three-dimensional structures of biomolecular moieties, like, for example, monoclonal antibodies and antigens that they bind to specifically. And what we can do with this kind of technology today is exploring the diversity of the immunome and the strategies that nature uses to generate the diversity and the specificities of antibodies that bind to recognize external moieties, like we did with the pangenome work, and understand what's the underlying language that is used by human genetics to generate the diversity of antibodies that are used to defend ourselves either from infectious threats or from oncological antigens, for example. Finally, the last bit that I would like to share with you is we have talked about manufacturing, we have talked about early discovery, and now I would like to mention another possible way of using artificial intelligence in the development phase, where you have to run animal trials and then clinical trials, and how you can do this in silico. And this is a multi-level simulation approach that starts with a natural language processing approach. We did this for RNA-based vaccines because of the reason I mentioned a minute ago. And the technology you use here, you start from a small seed of some scientific publications on a certain topic, and you derive a dictionary, and you feed this seed and the dictionary to a natural language processing machine that reads millions and millions of scientific articles in all the scientific literature and digests them, identifying those who contain relevant information for the system that you want to use. Through this digestion, the AI engine is capable of identifying the key experiments that have been performed, which are the experimental evidences, which are the key compartments of the system, which are the players in the system, and which are the relationships between these players. And basically what you end up with is a set of elements in a complex network, basically, that we depict with their biological and immunological interpretable symbols, but are just vertices and nodes in a network. And this network represents the mechanism of action of what happens immunologically once you inject an RNA vaccine in a muscle, for example, in your arm, and then from the arm it migrates into the lymph node, and then from the lymph node the antibodies and the BNT cells are produced, and they migrate to the bone marrow or to the blood. And this that you see here depicted as a static image, in reality it's a dynamical user interface that scientists can use to explore all the scientific evidences that are available on those topics and understand what's already known, what is not yet known, which experiments need to be performed to fill the knowledge gaps, etc. And this is the first application. Basically, it's human consumption. But the second application, which is even more promising, at least in my opinion, is that from this chart that you just saw, you can actually develop a mathematical model, because once you have a network and you have the relationships and you understand that the mathematical laws that govern these relationships, and you have experimental data that give you the parameters, you have a dynamic model that you can build. And from this dynamic model of the system that I've just described, you can actually simulate in silico what happens if you inject a certain quantity of RNA in a certain way, formulated with different options. And so you can perform in silico your trials to understand in advance which is the sector of the phase space which is more promising. And so you accelerate greatly by performing immediately the experiments in the areas of the parameters which bear the highest likelihood of success. And this is all I wanted to share with you today. I think we have still a few minutes for questions, and I'll be happy to take that.
Mr. Medini, thank you for a brilliant presentation, a brilliant lecture. Yes, we have a few minutes for several questions. And first, I want to ask you, what do you think will machine learning help us to create magic bullet for SARS-CoV-2? We see how artificial intelligence help in vaccine production, and what do you think in medication?
First, I'm a very skeptical person every time silver bullet is mentioned. I don't think that such thing exists actually in when you deal with science in general. Medications, therapies for SARS-CoV-2 or for viral infections in general are of two kinds. Again, they can be chemicals, and these are antiviral therapies, or biologics, and these are therapeutic monoclonal antibodies. Therapeutic monoclonal antibodies we already have. The problem we have the first one was the Regeneron cocktail, and then Eli Lilly manufactured others, and there are very promising late-stage clinical monoclonal antibodies which should get to the market in months. The problem with monoclonal antibodies is their horrible cost, and the cost comes from the amount of product that you need to administer to be efficacious. We are talking in some cases about half a gram or even a gram of biological product. It's an enormous amount. Consider that when we think of a vaccine, we are talking micrograms. So it's 6 orders of magnitude less. And producing those amounts is extremely expensive. The strategy there is to find more and more potent antibodies. The more potent the antibody, the less amount you need, and the cheaper the solution becomes. For antivirals, it's a different story, because the strategy of identifying antivirals goes through scanning, and artificial intelligence is very promising there, scanning huge libraries. But what you need to understand is what's your druggable target. And that goes through understanding the molecular level biology of the specific virus. We understand something about how spike opens, closes, and drives integration into the cell, but we don't understand everything. So that breakthrough is more related to an increased understanding of the pathophysiology of the virus.
Okay, thank you. Now we can switch off the presentation, and we have one more question from the audience. And what do you think about machine learning and artificial intelligence education? Do you think that nowadays every researcher, especially in the field of biology, must be educated and skilled in machine learning?
This is a very good question. I think the answer is no for the time being, because and I'll tell you why. I think every scientist in biology at large, biology, immunology, biomedical sciences, pharmaceutical sciences, in all this area should receive a strong training, fundamental training, basic training in mathematics. That's a must, in my opinion, a must that is not happening today. There's some Western schools, particularly in the United States, are going in this direction. Europe is not going in this direction as of yet. I'm not sure about Russia. It would be interesting for me to understand if this is happening in Russia at the moment. And before this happens, I don't think it makes sense to train in artificial intelligence someone that understands the application, but does not like biology, but does not have strong mathematical foundations. Because otherwise you end up, artificial intelligence is very, very tricky. You need to understand the fundamentals of statistical inference to avoid overfitting when you develop your model. And if you don't have those foundations, it becomes very risky to know how to use certain tools and how to apply them without having the foundations.
Thank you. And I think it's the last question. What do you think? Will the machine learning techniques replace the most of experimental science in the future, maybe near future?
In the near future, I think the direction is pretty clear. I think it's clear that there is what machine learning will, artificial intelligence in general, will replace is not the experiments, but it's the number of experiments that you do. Today, to reach a certain result, we need to repeat 1,000 times a certain experimental procedure. Artificial intelligence can tell you, instead of 1,000 trials, do this 10. And so you save 990 experiments. But you still have to do the final experiment in the living system on the bench to confirm the numerical prediction. In a fast-forward future, one may imagine that our ability to simulate the response of a living system could actually cancel certain experiments. And I've seen this happening in some specific, in diagnostics, for example. There are some diagnostics tests that today, if you do them with AI, you actually have a better accuracy than doing them with the wet lab experiment. But that takes a lot of experimental validation before you build the trust into the in silico methodology.
Thank you. Thank you very much for taking part for a brilliant lecture. I hope we'll see you soon in our future conversation and conference. Thank you, Professor Medini.
Thank you, Anna. It's been my pleasure, and I look forward to being with you again.
Thank you.